to share my experience with plant defense with you. So it is a very crucial topic. It has been crucial topic since long, but now it has taken a front seat as far as the crop protection or the yield and production of food crops is concerned. The evolution of crop and pests and pathogens, they have undertaken side by side parallelly, and the crops have been attacked by pests and pathogen since long, since the you can say that since the agriculture began. At in initial days, that might not have been a big issue because the demand for food grains and food products was less due to less population. But still, humans have devised various ways to protect their crop from pests and the pathogens. Obviously, the initial methods have been organic one, utilizing the natural resources or natural methods, and it went on smooth for say several centuries. But later on, with the advent of industrial revolution and other concomitant development, the population started increasing, and so the need for crop or the food products or food grains also increased. And so there was a need that not a single gram of food grain is lost due to infection. And that's why several
Your voice problem it होता है.
Sir, there is distortion in your voice.
it also involved, also not, it majorly involved the synthetic compound. And those
Voice
synthetic
issue आ रहा है, sir,
compounds,
please.
is my voice breaking?
Yes, bit little, but
कैसे?
not.
हाँ, sir.
Is it breaking?
Yes, sir.
Yes,
अभी
sir.
ब्रेक नहीं हो रही है, इसके पहले हो रही थी सर.
Sir, intermittently it is breaking, but now it's fine.
Okay, fine. And so there was a need that not a single gram of yield is lost due to the disease. And that's why people started using synthetic compounds to protect their crop. So these compounds, earlier they gave the benefits, or their benefits were only visible, not the side effects or the adverse effects. But then later on, slowly and steadily, we started experiencing or viewing their adverse effects on ecosystem as well as on the biota, and including the human beings. And so an interest in organic farming was again recreated, some say 2 or 3 decades ago. Still it is in infancy due to various reasons, but now people have been sensitized with the benefits of organic farming, although it has its own limitation. That is a different story. But still, now there is a need that we work on the organic alternatives for everything. So in today's talk, now since we are from a diverse background, those who are from a botany background, they must be aware about the plant defense mechanisms. But the others who are not that much conversant with the plant system, for them, I will be introducing the defense mechanisms of plant, various only an overview, not a very in-depth knowledge or information, just an overview, bird's eye view about the mechanisms of plant which it adapts to defend itself. And in next few slides, I will be concentrating on the economics of this disease. What are the outcomes? And there I will share you with some data that why there is a need to for disease management. And then lastly, I will be sharing my few experiments which I have done along with my student, by which we have established the induction of disease resistance, priming of plant with disease resistance using plant extracts. So this is a very basic thing which is called as disease triangle. Disease is always an exception. Host is always there. Pathogen is always there. But still, disease is not of common occurrence. It is just an exception. Even if pest or pathogen and host are available, unless they get the conducive environment, the disease is manifested. So only when these three factors meet, then only the disease develops. Now, whether the interaction between the plant and the pathogen or pest, I may be using the word pathogen throughout, but it implies to pest also. The interaction would be resistance or susceptibility. It depends on two factors. First is the substrate requirement of the pathogen. What is the need of the pathogen? What it requires for its survival, for its development, reproduction. And the second factor is the how plant respond to the pathogen. When the pathogen receives the when the plant receives the pathogen, what is its response? And based on the interaction of these two factors, whether the interaction will be susceptibility or resistance that is manifested. Now, based upon the substrate requirement of the pathogen, the pathogens are grouped into two groups, necrotrophs and the biotrophs. Necrotrophs, they derive their nutrition from the dead tissue of the host. So when they attack, they will release the toxin into the host so that the local tissue dies. If the plant is sensitive to the toxin of the pathogen. So if the plant is sensitive to toxin, it will kill the cell and the parasite, and the infection will occur. However, if the plant is insensitive to that toxin, so obviously that toxin will not kill the host cell, and the invading pathogen may not or will be unsuccessful in colonizing the host. The other group, that is the biotroph, it derives the nutrition from the live host tissue. And that's why it establishes the compatible relationship with the host. Necrotroph will develop the incompatible relationship, and that's why it will release the toxin. Whereas biotroph, it establishes the compatible relationship, thereby not harming the plant cell. It will send out its haustoria and absorb the nutrition from the live cell. The pathogen will be successful in infecting the plant by two ways. Either it should suppress the plant defense. Whatever response are being given by the plants towards the pathogen, those responses should be suppressed. Or the pathogen should divide quick enough so that it spreads throughout the host quickly, and the host doesn't get the sufficient time to kill the pathogen, invading pathogen, and thereby the biotroph will infect the plant. However, contrasting, if the pathogen fails to suppress the plant responses, or if it is slow in the growth, the plant gets enough time to kill the pathogen and evade the infection. Now, what would be the response of plant towards the invading pathogen? Either the plant should be resistant or should be opposite of that is susceptibility or susceptible. If the plant is resistant, it won't allow the pathogen to grow and reproduce. Or it would retard it to such a dead slow phase that till the pathogen develops or spread all over the host. The host has completed its life cycle. It has produced the required yield, and thereby the pathogen develops, reproduces, nothing negative or adverse effect will be there on the yield. So the plant has prevented itself by slowing down the pathogen growth and synthesizing the required amount of yield. Another way is that if host is unable to do anything, either stop the growth or retard the growth, then it will be susceptible. So here we understand the two ends, resistance and the susceptibility. Very clear cut. But actually, in nature, these two extremes are there, no doubt about that. But in between, there are several variations, right? So it may be mostly resistant or moderately resistant, mostly susceptible, moderately susceptible. So all sorts of variations lie in between these two extremes. Now, how the resistance is manifested by the plant? There are various ways, at various levels it can be manifested. First and foremost, it won't allow the spore to germinate. The plant won't allow the spore to germinate. So when the propagule itself is not germinated, there will be no question of colonization and spread and all that. That will be at the first step itself, it will be washed out. But in some cases, this may not be possible. The spore germinates, the propagule germinates, and it penetrates the cell. The plant kills the pathogen at that time, after germination, post germination, when the germ tube is trying to penetrate the host tissue. That would be the second step where the pathogen can be stopped. Even if that barrier is evaded by the pathogen, then the third is the plant will kill the pathogen during while it is in the plant tissues. It will kill by various mechanisms. We shall see those mechanisms also in the overview. So the plant will kill at that time. Even if pathogen evades this attack by the host, then the plant will adapt the mechanism by which it will slow down the pathogen growth or prevent the pathogen from reproducing. So at least this generation was infected. The next generation of host may not be infected if pathogen does not reproduce. So in this way, the resistance is manifested. So let's take one example of wheat stem rust, where all these mechanisms are there. Of course, the host and the pathover varies. There are three sets of genes which are involved in resistance. The first set of genes prevent the penetration of the pathogen. If these genes do not work, then the second set of the genes will be activated, which won't allow the development of haustoria. Haustoria is the structure by which the pathogen will draw the nutrition from the plant cell. And the third set of genes, which are called as the slow rusting genes, even if the pathogen is able to evade attack by all the mechanisms of first set, second set, there is third set of genes. Now these genes.
हेलो फ्रेंड्स, आपको सर की आवाज आ रही है?
ये मैडम बहुत
No, madam, it is breaking.
Yes.
Voice breaking.
बडेरे सर, हेलो.
voice is not clear, ma'am.
Yes.
Ma'am, voice is not clear. It is quite interrupted.
Yes, yes, yes. Just. हेलो. सर, वॉइस में कुछ इश्यू आ रहा है.
I think the signals over here are full.
ओके सर, आप वीडियो ऑफ रखिए, मतलब ऑडियो क्लियर आ जाएगा. आई थिंक वीडियो ऑफ
ठीक.
रख कर दीजिए.
I will have to go out of the presentation. Please
नो
give me.
सर, नो सर, इन द हेडर रीजन आप थोड़ा कर्सर ऊपर लेके जाइए
अच्छा.
स्क्रीन की तरफ. वहाँ पे ऑरेंज
Okay.
कलर का आपको स्ट्रिप दिखेगा, उसमें है ऑडियो वीडियो का ऑप्शन.
Okay, okay, okay. Okay. Yes. Stop video. Okay. So this was an example where you can see that there are various ways by which the host plant evades the pathogen attack. Now the question arises, how does the plant recognize the pathogen? So there are a set of molecules called as elicitors. Now these elicitors can be anything. It can be some derivative pathogen. It can be the derivative of host. Mostly they are oligosaccharides, the components of cell wall of the pathogen as well as the host. Now these elicitor molecules, they trigger the signaling mechanism in the plant by which the host is able to recognize the pathogen. For example, when pathogen attacks the host, it will have some molecule around it on the cell wall. So these molecules are first recognized and then trigger the defense responses from the plant. Plant produce enzymes which degrade the cell wall of the invading pathogen, and when this occurs, the degradation product of the cell wall, they themselves act as elicitors. Now, when the pathogen has to enter inside the plant cell, it also releases some hydrolytic enzymes. These hydrolytic enzymes degrade the plant cell wall of hemicellulose, pectin, and all polysaccharides. On degradation, these polysaccharides produce oligosaccharides, which again prime the host cell. So thereby, a series of interaction occurs, several more, and this is how the host is the pathogen. So these elements of very two broad categories are non-specific all call it as an can also call it as a defense response. So these non-specific elicitors are cell wall fragments, peptides and other glycoproteins, and polyunsaturated fatty acids, component of the cell wall and membrane. So this gives the generalized defense priming to the plant. It is called as non-specific because the defense priming induced by elicitors from one pathogen can act on a series of other pathogens also. Not only that, some molecules, chemical agents, physical agents, they also trigger these mechanisms, like say heavy metals, mercury chloride, copper sulfate, UV rays, all these chemical and physical agents, they trigger these mechanisms. And they will provide, as I have told you, the generalized defense priming. Against a range of pathogens, resistance will be developed in the host tissue. And then there is gene specific elicitors, which are coded by the AVR gene in the pathogen. AVR stands for avirulence. For every product coded by an AVR gene, there will be a corresponding R gene in the host. So when this recognition occurs between AVR, product of AVR gene and product of R gene, then there will be resistance. Otherwise, there will be susceptibility. So this is a very specific mechanism. It is so specific that for every race of a pathogen, a separate R gene is there in the host. So the resistance induced by one pathover of the pathogen is not active against the the different pathover of the same pathogen in the same host. And that's why it is called as the gene specific elicitor. As there are elicitors, there are suppressors also. Mostly biotrophs produce this. These are chemical biotrophs. They produce suppressors, and the plant is unable to respond to the invading pathogen. So this suppressor will result in delay of the recognition of pathogen by the host species, or the host species may not at all recognize the pathogen. Like the certain Ascochyta rebi, it produces certain glycopeptides in which are active in chickpea while it is colonizing the chickpea plant. Okay. Now we shall have an overview of various mechanisms of the plant which they employ to defend them against the pathogen. And if you see, you will find several parallels between the animal defense mechanism and the plant defense mechanism. Like you can group them into two groups, passive and active. Passive or innate mechanisms, they are always there, whether there is a pathogen attack or not. Active mechanisms, they are activated only when the host is attacked by the pathogen. So under pathogen will attack, try to gain entry into tissue. Below that, there will be cuticle, which is secreted by the cell itself. Cuticle has several functions. It provides desiccation, but one of the function is also to prevent the pathogen entry into the host tissue. It gives a barrier to the invading pathogen. The same function is carried out by the cell wall. Chemically, the cell wall will be the same, but physically it will be different. Some cell walls are strong, some are weak. Some have a thick network so that the pathogen is unable to get easy entry into the host tissue. Where the cell walls are weak, the pathogen has a free pass into the host tissue, the protoplast of the cell. Then there are natural openings on the plant, like stomata for gaseous exchange, lenticels, which are produced due to the secondary growth. The epidermal tissues, they break and the lenticels are formed. So these are natural openings. Although they are not barrier as such, but like for stomata, since it is an opening, it is easier for a pathogen to gain entry into the tissue. But there is one example in coffee. The resistant varieties of coffee have smaller stomata. Now, since the stomata are small, the larger spore cannot enter inside the coffee plant through this stomata because of the size difference. Susceptible cultivars, they have larger stomata, and that's why the pathogen can enter. Similarly, the lenticels are there through which the pathogen can enter, but what plant does, it will plug that opening through the development of tissues like cephalotory tissue, which has say hydrophobic compounds. The water is not there, or it is water hating compounds are there in those cells, and that's why the invading pathogen infect the plant. It is whether it is a necrotroph or biotroph, it is for the nutrition, for its growth and development, for its sustenance. If the plant refuses to provide the required nutrition to the pathogen, then the pathogen will be killed there and there. It won't infect, colonize the host. So the resistant cultivars, they do not have the nutrient which is required by the pathogen. Then there is pH. pH is important because pH affects the chelation of the ions. Some ions which are required, necessary for the growth of pathogen, the chelating compound at the appropriate pH will chelate those ions, and thereby the pathogen dies of starvation. Then there are phytoanticipins and the plant defensins. These are the antimicrobial compounds produced by the plant. Resistant cultivars, they have the capacity to produce phytoanticipins and plant defensins in a larger amount, thereby excluding the pathogen. The susceptible ones are not capable of producing enough of these compounds. The active mechanism, as I have told you earlier, will be activated only after the pathogen attack. So there are certain mechanisms which are activated within few minutes of the pathogen attack, and those are grouped as rapid responses. And then there are responses which take few hours to few days for development. So rapid response presence of oxidative species produce the hostile conditions in the cell. Although the plant cell will be killed, no doubt about that, but it will also kill the pathogen. So it is a sort of suicide prevention. Few cells will be killed to prevent the rest of the plant from the pathogen attack. Then there is cell wall reinforcement. Pathogen has gained entry into the cell. It has broken the cell wall and gained the entry. So that broken cell wall is repaired quickly by the plant so that the other more pathogens do not gain or the secondary infection does not occur in. Then there is hypersensitive cell death. Again, it is a mechanism to prevent the spread of the plant growth. Oxidative burst and hypersensitive cell death, they both are related. So oxidative burst lead to the hypersensitive cell death. So it is also called as HR, hypersensitive response. Then plants also produce phytoalexins. These are the antimicrobial compounds, low molecular weight, mostly phenolic compounds, which will kill the pathogen. Then in delayed response, most of these responses are local. Local means they will occur only in the tissues which are directly infected by the pathogen. Then there are some systemic responses also, which develop the systemic acquired resistance. So these responses prepares the plant for pathogen attack. The tissues there, they are not under the direct attack by the pathogen, but wherever the infection has occurred, from there the signals they are generated, they spread throughout the plant and probably to the nearby plants also, and prepare those tissues, those plants for the pathogen attack. Now this systemic acquired resistance enables the plant to respond quickly against the pathogen attack. Then there are certain PR proteins or the pathogenesis related proteins which are involved in development of systemic resistance. Then there some PR proteins are also involved in containment of the pathogen and all that. So these are the overview of the mechanisms adapted by the plant to defend itself against the pathogen. Now, before I have come to my topic, let's have some what you can say, a look at some economic considerations of the crop diseases. The most of the studies in the plant research they have been carried out in crop plant. That's why we have most of the information. Is there any problem? Can everyone hear?
No, sir, your
The
voice
voice is
Hello.
interrupted.
Hello, sir.
Voice
Your
is cracking.
voice is not proper to come.
Okay, okay.
Some disturbances in that.
कभी कभी बहुत अच्छे से सुनाई देता है लेकिन कभी वो और भी धीरे होता है.
Acha, can you hear now? Hello.
Yes, sir. Yes, sir.
Yes, sir.
It
Okay,
is audible now.
okay, okay. Now, all these studies have been carried out in crop plants, and that's why, and that is only because of the agriculture. We are dealing with the loss of yield due to diseases, and yield is an important factor of the crop. All other characters come later on. No doubt, or no matter whichever variety you develop, unless it is high yielding, the farmer is not going to cultivate that variety. Because what farmer is concerned about is the yield. Therefore, it is how the economy works. This yield is a very complex character. There are several factors
are involved in developing, resulting a particular yield in the crop. Physiological characters are there, morphological characters are there, molecular characters are there, environmental factors are there. All these factors together, they decide the yield. With the scope of this lecture, we will be concentrating only on the biotic stress, an important factor affecting the yield of the crop. Now, since say last 2000 years, you can see from this graph, in recent year only, the agriculture area has increased enormously in world over. Right? So far, say around 1580, the area under cultivation was more or less same. It didn't affected much. And after that, you can see there is a steep increase. Mostly after 1700 AD, there is a steep increase in the area under cultivation world over. And this is the period which coincides with the Industrial Revolution, by which the population started increasing and the demand of food also increased. If you last 60 years, you will see that the agricultural land per capita has decreased enormously. So from somewhere above, say for world over, it was about 1.5 hectares per capita. It has came down to say 0.6 or 0.7 hectares per capita, almost half world over. It is a world average. It varies with to continent, but if you take world average, it has come down to half. Similarly, the agricultural land initially, with the development of civilization, if you look around 7000 BCE, it went up steeply, and here you find several civilizations developed. And then it was the increase was not that much, and then there was a plateau for a sufficient amount of time. And since the last 2 years was say last 2000 4000 years, there was a steep up, and it was again reduced drastically in last say 50 years. However, if you look at this graph, then in last 60 years, the yield has increased. Yield of various crops has increased enormously. At least there was increase in the yield of at least 50%. In crops like wheat and maize, it has increased more than 200%. Now, in spite of enormous decrease in the agricultural land per capita, we are not facing that much of food scarcity as we could have, only because there was a parallel increase in the yield of the crops. That simultaneous increase in the yields of the crop have saved us from the starvation. Now, this is a graph which shows the land which is used for cereals. Cereals are the main food source. So, world over, in say for example, here the cereal yield has increased, land have increased together. But in Asia, where majority of the human population reside, land use for cereal cultivation has not increased much. Not increased much. But yield has increased enormously, more than say almost 250%. And this is one more evidence that increase in yield has saved us from starvation. Now, how could we achieve that much enormous yield? There are various reasons. No doubt, we have produced high yielding varieties, resistant varieties. But one major factor which has contributed in the increase in the yield is the use of synthetic fertilizers. So see, if you look from say late 19th century to early 21st century, there is an increase in the application of fertilizers. Recently, we see some drop, not world over. The world over trend is still over applying the synthetic fertilizers. And this is also evident from the increase in the consumption of fertilizers since last 60 years. It has increased from 50 million ton to more than 200 million ton. And the use of fertilizers for agriculture is more in the developed countries or developing countries rather than the underdeveloped countries. Where the income is low, people do not use fertilizers. Only and lower middle and higher income range is use of fertilizers. And mostly we use nitrogen and then phosphate and the potash. And this shows the trend of using fertilizer per hectare. See, China, Egypt, they are leading. Some part of this Arab also, this Eastern South America. Here also, the fertilizers are used mostly. Even in India, we are using much amount of fertilizers. Now, these fertilizers completely are not used by the crop. They remain. We are using more than required by the plant. And that's why that excess either remains in the soil or it is washed off, thereby polluting the agricultural land, water bodies, and the complete ecosystem. Here you can see this is an example of excess of phosphorus from the crop. See, India is on the higher side, India, China. These are on the higher side. So they use enormously. Now, sorry. World population has increased logarithmically in last say 100 years. There is good amount of population in the world which is not supported by the synthetic fertilizer. But again, it's not much. Population which is supported by the synthetic fertilizer, it is increasing linearly. Now we come to the comparison between the agriculture which uses synthetic compounds and the organic agriculture. And before 20 years, there was hardly any land which was under organic cultivation world over. This is all the world average. Now it has started little bit increasing. As the population which uses say synthetic compounds, it has remained stagnant. Now we come to the impact of diseases on the production and yield of the crop. It is an estimate that worldwide there was a production of $1.5 trillion. It was expected in 2002. But actually we could achieve only 950 billions, means only 63%, 63.5% of the expected production was achieved. And there was a loss. There are many factors for the loss. Out of that, $220 billion loss was because of the pathogens, which is about the 14.1% of the production. We could achieve $455 billion of production. Out of this, only $450 billion production was prevented. So this is the impact of the diseases. If we are able to prevent this loss, it adds up to our production and the yield of the crop. So the pest or pathogen, it attacks the crop, which has several effects, and one of the effect is the yield is lost. Yield penalty is there. When yield loss, people spray synthetic chemicals, insecticides, fungicides, so that there is no infection and the yield loss is prevented. But these synthetic chemicals, they pose the threat to ecosystem. They create pollution, not only in their use, that farmer will spray or apply, which will contaminate the soil, which will contaminate the water bodies. At the same time, when these chemicals are being manufactured in the factories, those factories also generate pollution. So the pollution is at every stage. Extraction of that crude organic compounds, synthesizing the chemicals of interest in factories, and then using it in the farmer's field. So at every step, the pollution is generated, it is created. So our worry is about the pollution. No doubt, we need yield, but also at the same time, we do not need pollution. So this is the which shows you the trends of using pesticide. It is being used world over, and where you do not see there is hardly any agricultural activity. And most of these chemicals are in form of say fungicides, insecticides, bactericides, herbicides. So what is the alternative? First is the resistant varieties. Use of resistant varieties. It is an ongoing process. Breeders will breed resistant variety. They will work for few years, and later on, as the pathogens which could breach the resistance of these varieties develop, the resistance will be lost, and those varieties will become susceptible. Eventually, they will become susceptible. So this is not a permanent solution. Organic farming, okay, it is a good alternative, but if you compare the cost of the products organically, which are organically produced and which are not produced organically, there is a vast difference. Say a rice which you may get 60 or 70 rupees a kg, the organic rice, the price will be 250 or more than that. And that price is not affordable for the common man. Only few can afford that, but all cannot afford. So at present time, this is not a commercially viable alternative. Now, third option which is available is the triggering plant's defense mechanism. Somehow priming it. It is like vaccination. We have just most of the human population has vaccinated themselves against COVID-19. So you trigger the defense mechanism in the plant so that the plant is already primed, a systemic resistance is produced, and whenever there is a pathogen attack, the plant will defend itself more efficiently as it could have in the absence of priming, and thereby reducing the yield lost. So how you can trigger this plant defense mechanism? Now, since I am concentrating on the organic alternative, there is synthetic alternative to this also. There are certain synthetic chemicals which are available in the market which trigger plant defense. But those are synthetic ones, not the natural compounds or natural organisms. Technically, you can use two methods. First is the use of PGPRs, that is plant growth promoting rhizobias, and plant extracts. Both of these can be used either alone or in combination. So what these PGPRs do? They will use lytic enzymes which hydrolyzes the cell wall of the pathogen, thereby killing it. It enters into a phenomena called as antidosis. It won't allow the pathogenic bacteria and fungi to grow, thereby excluding them from the rhizosphere of the plant and preventing the infection. Then these PGPRs also releases siderophores, and this siderophores molecules which will chelate the essential elements in the rhizosphere, depriving the pathogen from those ions, and the pathogens are starved to death. This is how the PGPRs they act. And plant extracts, they have oligosaccharides in them. They have other compounds also. Trigger the defense mechanism in the plant and induce the genes which are involved in plant defense. So these are few reports where the PGPRs and plant extract they have used separately or in combination. Now with this thing in mind, now in last say I think only 20 minutes are left. In this 15-20 minutes, I will share my work with you. So we were our aim was to look for some green alternative for disease management, and that too from the local flora, so that it can be what you can say exploited by the local people. They don't have to go to some multinational company which will sell them at a higher prices. They can use those plants from their nearby vicinity. And then we have published a few papers, few we have published, and one or two papers they are on in pipeline. So we have studied the defense mechanism in cucumber, that is Cucumis sativus, and Capsicum annuum, is chili. Both are important vegetable crops. And we have studied the induction of systemic resistance in the plant by studying the activity of marker enzymes, kinases, glucanases, peroxidase, and the catalase. And then we have also studied synthesis of antimicrobial compound by the plant, synthesis, where the phenylalanine ammonia lyase, it is the key regulator enzyme of the phytoalexin synthesis. Chitinases, they degrade the chitin and produce oligomers, dimers, and the monomers. Glucanases, they cleave beta-1,3 bonds. Then what we have studied is the beta-1,3 glucanase. Then there are beta 1,4 glucanase also, which cleaves the 1,4 linkage. Well, as I have told you, it what you can say deaminates phenylalanine and produces transenamic acid, the first compound in the synthesis of lexine. Through the series of reactions, produce several sorts of phytoalexins. Then the peroxidases, these are the antioxidant enzymes. Although oxidant molecules are required for the killing of pathogen, but still that excess quantity has to be kept in check. So under pathogen attack, when higher amount of antioxidant reactive oxygen species are produced, the activity of oxidase it increases, as well as the activity of catalase also increases. So what we have done, we have elicited the ejaculated seedlings of these plants with either a known substance inducer or the plant extract, and then assayed the activity of the enzyme to establish that the disease resistance can be conferred to the plant by the use of plant extract. So for growing the ejaculated seedlings, we have surface sterilized and then allowed to germinate and grow under the aseptic conditions in dark for say 15 to 21 days, depending upon the crop. Initially, it was necessary to determine when the peak activity of a particular enzyme occurs, right? So that is very much important for assaying the activity of the enzyme. So initially, in initial experiments, we spread the seedlings with known elicitors like UV rays, then copper sulfate, mercury chloride, and all that. And then after spraying, we fix those seedlings in liquid nitrogen from 0 to 75 hours of elicitation. At the interval of 5 hours. And subsequently, we determine activity in various tissues like cotyledonary leaf, hypocotyl, and the root. So these enzymes we have said, as I have told you earlier, and then protein quantification was Bradford method. This is one of the representative graphs which tells what we wanted to know from these experiments. If you see unelicited control, here the enzyme activity was more or less similar throughout the experiment, 0 to 75 hours. But elicitors like ultraviolet rays, mercury chloride, copper sulfate, salicylic acid, and fungal cell wall elicitor, you will see that initially there is hardly any significant change in the activity. Later on, the activity starts increasing slowly and steadily and reaches its peak around 65 hours, and then it subsides. So this peak is necessary. This is the region where we should assay the enzyme to look for the effect of the elicitation by the plant extract. So after knowing that, we screened the plant extract which was active, which had the ability to induce systemic resistance and synthesis of phytoalexin in the seedlings. So we selected natively available plants, particularly tree species, and that to the foliage part, because these are the regenerative tissues of the plant. We don't have to sacrifice the complete plant when we take the few leaves from the plant. The plant still remains there. And that's why our interest was only in the foliar parts of the plant, like neem, Calotropis procera, what we call as rui, Clisthanthes spolinus, that is called as gamari, Sisam, Dalbergia siso, Datura inoxia, and Lantana chimera, what we call as haldi kumku. Right? So we took the aerial part, dried it, powdered, and then extracted in water. We have used fully the water as a solvent for extraction, not the organic compounds, because water is available everywhere and it is almost free of cost. So the farmer doesn't entail any cost on extraction also. Although the farmer cannot centrifuge, but the farmer can filter extract so that the particulate matter is removed. For our studies, autoclaving was required only to rule out the possibility by elicitation by the flora, the microflora which was present in the extract. In farmer's field, this is not required. So we have the volume, and the range of concentration varying from 2 to 20%. So this extract was sprayed over the seedling, sterile aticulated seedling, and then fixed in liquid nitrogen at 0 hour elicitation, and at 3 hours, at the time when peak activity of the concerned enzyme occurred, 5 hours before that and 5 hours after that. Acid enzymes, as I have told you earlier. So again, this is one of the representative graphs I want to share with you to understand what we have done. So we have fixed at 0 hour. The kinase activity was at 65 hours. So we have fixed at 65 hours, 60 hours, and 70 hours. So here you can see low concentration was hardly any elicitation. Higher concentration of say 16%, 20% over here have induced significant increase in the activity. Maximum was with 20%. So more or less the similar case was with all other plants and all other tissues. So we have selected 20% extract for further study. So this has established that 20% extract of the plant induces maximum activity. Yeah, this is the example of Clisthanthes spolinus. From our study, we found that these two plants, neem, Azadirachta indica, and Clisthanthes spolinus, they induce maximum activity. Rest also induce, but their effect is not that much consistent and not at that magnitude also. So only to give you an idea, let's compare the activity induced by 20% extract of all the plants in all the tissues. So in cucumber, with respect to chitinase activity, you can see Azadirachta indica and Clisthanthes spolinus have induced activity in all the tissues, whereas like Dalbergia, Datura, and Lantana, they have induced only in roots. Procera, none of the tissues. The glucanase activity, you can see, is induced by Azadirachta indica, Clisthanthes spolinus. Here, Rupesh Procera has induced in cotyledonary leaf and hypocotyl, and in other plants they didn't induce much. Significant. The same thing is for the peroxidase. Here, Clisthanthes spolinus has induced, Procera has also induced, Calotropis, as well as the Lantana has also induced. Catalase, Clisthanthes spolinus, Azadirachta indica. If you look at such, all the tissues induce the activity of catalase by the extract of all the plant. Again, major was from Clisthanthes spolinus, Azadirachta indica, and occasionally from Calotropis procera, and Lantana camera. So the findings in Chile, here again, Azadiracta indica, Clestanthus, Calotropis, and the Lantana. For glucanase, Azadiracta, Clestanthus cholinus, Futura, some sort, in Dalbergia siso, and Lantana camera also. For oxidase, Azadiracta, Calotropis, and the Clestanthus. Catalase, again, in most of the tissues, the activity was appreciably induced by almost all the extracts. So overall, you see that maximum effect was either of Clestanthus cholinus or Azadiracta indica. But since Azadiracta is already characterized, we concentrated on Clestanthus cholinus because it was a new report. No one had reported till now that such activity of Clestanthus cholinus. Right. So the also. Now, then we tried to find out which fraction of this extract is active. So in a very crude way, we have fractionated this extract over the silica column and then diluted it with 95%, sorry, 90%, 45%, and 0% of methanol. And each of these fraction was reconstituted and used for elicitation. So you see that we have applied 1.75 grams of extract. And through this column, we could recover 1.7 grams. That means almost all of the extract was recovered from the column. Then we have sprayed the seedlings with plant extract, fraction 1, fraction 2, and fraction 3. That means fraction derived from 90% methanol, fraction 2, 45% methanol, fraction 3, 0% methanol. That means only water. So here you can see that the fraction 2 and fraction 3 they were not mostly active. Fraction 1 was active with the plant extract. That means all the compound which are active in inducing plant defense is present in the first fraction of the extract. So these were the, what you can say, findings in almost all the cases. So I'm not going to show you all such graphs. So we could find a similar result in every aspect. So later on, just out of inquisitiveness, scientific inquisitiveness, we tried to see the effect at molecular level through isozyme analysis. Since the fraction was only active, we have used only plant extract and fraction 1. We have studied the zymograms of kinase, glucanase, peroxidase, and catalase. So we fixed the seedlings, elicited seedlings in the liquid nitrogen, then extracted, electrophorus over the native gel. It is important. We cannot run SDS page over here. We have to run native gel. Then incubation with substrate, and then development and visualization. In cucumber, you can see in cot leaf, this is the profile in control, and elicitation has modified this profile. Here you can see an extra and over here. One which is seen in is not seen in illicit seedlings, but new to as they appear. Similarly, in root also there is change in the pattern of expression. In glucanase, there is a slight change in ciliary leaf, no change in hypocotyl, a significant change in root, and the two between one extract and the fraction 1. peroxidase, here you can see these are the extra band which are synthesized. Similarly, these are the extra band synthesized in response to elicitation. The same in hypocotyl as well as in the hypocotyl. No change in the root. Catalase also you can find mostly in the quantity rather than the size of the isoforms. In fully, again you can see there is a perceptible change in cotyledonary row and the root, and no change in hypocotyl. In glucanase, we find an extra band in section 1 in cotyledonary leaf, in root, in plant extract, as well as in the elicited with the fraction 1. Same story goes for peroxidase as well as for the catalase. So you can see that the elicitation has resulted in change in the expression of the isoforms of the enzymes also. Then we have just had a very crude analysis, chemical analysis of the extract. So section 1 is, say, rich in phenolics, right? If you live, it goes for the plant extract. But if you compare amount of phenolics extracted in fraction 1 is more, right, compared to the extract. Similarly, the percentage is also more, as well as here you can see the flavonoid. So all these compounds, phenolics, and carbohydrates, flavonoids, these are proved to be involved in triggering the plant defense mechanism. And here we are getting maximum concentration in the fraction 1. Qualitatively, we have done TLC analysis. Here also we have found what you can say improvisation in the pattern of fraction 1, which in the with respect to the compounds which are involved in the triggering of the defense responses. So probably in our case, from this study, we can say that all these compounds or few of these compounds may be active in triggering the plant defense responses. Since the treatment with Clestanthus cholinus resulted in the expression of markers of SAR and the key regulator of the phytoalexin metabolism, it can be used in field for disease management in the crops. But however, a detailed study is required, mostly with respect to the biosafety of the extract. Now, she is Dr. Monal Kru, who has carried out this study. She is my student, and she has conducted all these painstaking studies which I have presented here. These are part of the studies which I have presented. There is other studies also have been done, but with the scope of this lecture, I have presented a part of her thesis with you. I have shared with you. So these are the references, and probably you must have enjoyed listening to this work, and I was successful probably in conveying my message to you. So it's over from my side.
Thank you so much, Dr. Badere, sir. If anyone has any query or question, please ask.
Good afternoon, sir.
Yeah.
Sir, I don't have any query as such. Just I wanted to share that one of our students in the department has worked on the project on the similar lines.
Okay.
Like what he took the waste flowers from the temples. Like in temples, we have lost lots of different types of flowers which get accumulated.
Yeah.
and they go to waste. So he took the different combinations of, means whatever temple waste was there, he collected and he prepared the extracts.
Yeah.
Then he prepared the spray from it.
Okay.
And he used it on some plants and some vegetables. Like few I remember is like on garlic, the spoiled garlic. He applied the spray to it, and then onion, spoiled onion, and also on apples.
Mhm.
So he got very good positive results.
Yeah.
It
Very
was a small, not a PhD project. It was a MSc by research project. So he
okay.
did that much only.
Yeah. Nice to know that.
Mhm. That much, sir. Just now he completed his degree.
Yeah. So have you published that work?
Yes, sir. He has published with his guide.
Okay.
I think partly he has published. The last results which he got, I think that have not been published.
Okay. Okay.
Any more question?
Hello.
Yeah.
Very good afternoon, sir. Sir, a very excellent presentation. Sir, I do have a query. Actually, I'm working on BT cotton pest. My PhD work is based on Spodoptera luteura by control measure. And actually, I used a plant extract, methanolic extract to control them. Apart from that, sir, in case of like a natural pest enemies and all, so what do you think about the Chrysoperla carnea to use as in
So,
pest control tool?
I'm unable to understand your question. Please speak a little bit loudly and slowly.
Yes, sir. Sir, actually, I work on Spodoptera luteura pest management.
Okay.
It is a tobacco leaf eating caterpillar.
Yeah.
To control pest menace, we had used a certain biological organic extract like plant extract of Lantana camara,
Yeah.
Osmum santum, Azada
Yeah.
ricta indica.
Yeah.
But apart from that, sir, as a, again, as you said that key as a part of organic control, apart from using this plant extract, we can
Hmm.
also go for that pest in terms of that natural enemies. We can use it.
Yeah.
So what whether we are quite capable to make hostile these natural enemies in a laboratory environment?
Means you want to culture those
Natural
those
enemies,
bacteria.
yes. No, sir, it is natural enemies like we are in chrysopedes are over there. Even though Chrysoperla carnea,
Okay.
then
Okay. Okay.
there are certain hemipteran bugs are there. So on how we can manage to culture them in laboratory? Is there any
Uh,
kind of other methods?
Sorry, I'm not an entomologist, so I have no experience in rearing insects. But
Right, sir, right.
yes, probably it can be done because people are doing. They are rearing the natural enemies, and then they are releasing in the environment.
Right,
So
sir, right.
the entomologist may be probably best person to answer your question. What we are doing is what we are not directly killing the pathogen, right?
Right, sir, right, sir.
Or the pest. We are only arming the plants with defense mechanism. Whether they kill or exclude the pathogen or pest, it is up to the plants.
So it's a natural defense mechanism.
Yes, we are only triggering the defense mechanism in the plant. We are not directly killing the pathogen or the pest.
Right, sir,
Right?
right, sir, right.
So in the whole study, you can see that we have only concentrated on the host responses.
Right, sir.
So that is the difference between probably what I could understand your way and the way in which we are working.
Right, sir. Thanks, sir.
Hello, sir.
Yeah,
Hello, sir. This is Kushal.
yeah.
Sir, I have got some experience regarding this Clistanthus colinus plant.
Yeah.
We have seen some farmers were using Clistanthus colinus plant leaves in their fields, paddy fields.
Okay.
So today we came to know that it's a scientifically proved that it elicits the disease and it controls the fungal disease. So
Yeah.
I think your work is very nice, sir, and we came to know something the scientifically proved facts we have seen in your presentation.
Yeah.
So thank you for sharing your nice piece of work.
And I also thank you for validating my findings in the field itself. I was unaware about that. Right? Thank you, Dr. Borkar, for updating that to me. So probably there is no biosafety concern which I was worried about Clestenthus cholinus.
Something it is said that that plant is toxic.
Yeah.
That's why many people do not use it openly.
हाँ.
वो जहरी है, जहरीला है, इसलिए उसका इस्तेमाल नहीं करते, जहरीला समझते हैं उस प्लांट को.
People use their leaf for fish poisoning.
Yeah.
हाँ, ये तो जाहिर है कि वो पब्लिक जो है छोटे से पॉन्ड में इसके जो पत्ते हैं क्लिस्टांथस के वो पॉन्ड में डाल देते हैं फिश को मारने के लिए, और फिर वो मरने के बाद फिश उठाकर ले जाते हैं. तो इसलिए वो फार्मर जो है वो उसको ओपनली यूज नहीं करते, वो चुरा कर फील्ड से लेकर आते हैं और अपने खेत में डालते हैं. इस तरह से हमने देखा है.
Okay. चलो, nice information. Thank you.
Thank you, sir.
Welcome, sir.
Anyone else? Okay, so thank you so much, sir, once again. On behalf of HRDC Nagpur and from all the participants, I extend my thanks to you, sir. Thank you so much.
Thank you, ma'am. And thank you, Dr. Dongar.
Thank you, sir. Thank you all of you. यहाँ पे अपने सारे सेशंस खत्म होते हैं आज के. We will meet tomorrow at 7:15. और जिन्होंने वेबिनार का रजिस्ट्रेशन नहीं किया होगा, कल अपना वेबिनार है 10 से 11. Have you all registered?
Yes, ma'am. We have.
Yes. तो देखिए आज रात को हाँ ओके जस्ट अ मिनट. आज रात को जो मैं वेबएक्स की लिंक शेयर करूंगी वो अपने मॉर्निंग योगा सेशन के लिए और आफ्टरनून जो 11:30 बजे से आगे सेशन है उसके लिए रहेगी. और अपना कल का योगा सेशन होने के बाद वेबिनार के लिंक में शेयर करूंगी. उसका नाम अलग रहेगा, वेबिनार का नाम उसमें रहेगा, तो डोंट गेट कंफ्यूज. आर यू गेटिंग?
Yes, ma'am.
हाँ, तो थोड़ा सा लिंक देख लीजिएगा जब आप ज्वाइन करेंगे तब.
Okay.
So thank you all of you.
Thank you, ma'am.
Thank you.